Good morning, good afternoon, good evening, hello to wherever and whenever this is finding you. And welcome to lecture number 2 of Zoology 2700. Previously on 2700, we talked about the course. We kind of advocated for why I'm excited. Well, I demonstrated why I was excited about the invertebrates of the world, advocated for you to also be excited and to go out and evangelicalize about them as well. And today, what we're going to do in our time together is to essentially agree that we're on the same page. Not everybody has taken the same array of courses to get here. One of the common ones that you ought to have had by this point is some thinking of about evolution and some familiarity with evolutionary terms. And so beyond just thinking and presuming that we're all sharing these terms and sharing these experiences, what I'm going to do is to talk about some of what I think are going to be the most used or most useful to you evolutionary terms, particularly with regard to phylogenetic systematics and to tree thinking. And we're going to go through one classic example that probably you've been exposed to in other courses and potentially even in your high school training, but earlier in your career as a biologist, in terms of evolution by natural selection. And we're going to claim it here because it's a classic example that is utterly dependent upon invertebrates, because it's this picture here. It involves the pepper moths in the United Kingdom. We're going to then finish up by preparing for the jump that we make into the eukaryotic world, and we'll talk about serial endosymbiotic theory and the evolution of eukaryotes. So these are the learning objectives, from the learning objectives in the core syllabus. This is what I believe we're talking about today, demonstrating a solid understanding of basic evolutionary principles, and then beginning to, and remember these active verbs, appraise, synthesize, and interpret examples from simple phylogenetic trees. Fair enough? Here we go. I pause. This is a red herring, not a red herring, but this is an aside already. One of the things I'm going to ask you to do throughout our asynchronous times together is to pause me. All right? You've got that power when we're asynchronous. And so there's a couple of things in today's lecture where we're going to actually actively ask you to make a prediction. I'm going to show you some graphs. I'm going to show you some axes. Pardon me. And then I'm going to ask you to make a prediction. So pause. Do that. Go through the exercise. Is play along, because getting things wrong is a really important part of learning, right? And there is zero consequence here in this asynchronous world. So play along, and as I ask you to make some predictions about what trends you would see in a graph, play along. And use the pause feature, especially if you have me talking at 1.5 times speed, just so that you can get through more of these together. So, all right, we agree? Let's begin with housekeeping. And one of the things that I'd like to remind you about is this request that I made for you to go to this Google Sheet and find your student number in this column, the first column, and then put a 1 for whatever column of the phylum that contains your favorite invertebrate species. So if it's a nudibranch from underneath the ocean, gorgeous one off Papua New Guinea, find mollusk and put a 1 under that beside your name. All right, we'll use this. I'm going to collect this kind of 2 times in the year, and I'm interested in potentially the changes through time. So this is the Dunn phylogeny. We talked about it briefly the other day on Monday in our shared time together, and we're going to revisit this, particularly as we leave the protozoans. That's kind of the first taxa that we're going to talk about, are the protozoans. That'll happen together on Friday, or begin to happen on Friday. So you'll see more of it into the 2nd and 3rd week of January, and then for the rest of the term. But we're going to talk about it today as an example phylogeny, because we're going to be talking about phylogenetic terms. So this particular one comes from a decade ago in the Annual Review of Ecology and Systematics. You can scan that QR code and go see the actual literature, the paper, if you'd like. And this is also an encouragement, if you've got the text, the materials we're talking about on Monday, Wednesday, and a little bit onto Friday, you can brush up on some of the thinking in chapters 1 and 2. Now, one of the terms that we're going to unpack later on in today's lecture is a polytomy. And so I'm going to kind of foreshadow that by giving you one here and say the spiralia, this little dot here. You can see that there is not a dichotomous, like a branching of 1 to 2. This is a dichotomous branching here between Ento and Procta and the Cycliophora. But within the spiralia, the relationship between the taxa contained within the spiralia is uncertain. This is not a knock on that phylogeny. It's a statement of our knowledge of the world right now, and a statement of the resilience of this tree, because we can take this tree with its 34 tips. I'm going to flip it on its side and then say, here's that yellow dot again. There's the spiralia, and I've collapsed those uncertainties. And I'm encouraging you, here I am encouraging you to look at this and say, there's still a lot of information here, and we know there's still a lot that we know and can unpack in the world because of the simplification of this tree. And so looking at the bilateria, there are deuterostomes, spiralians, and ecdysozoans. That's a valuable group for us to look at because there are some traits that are generally possessed by all of the taxa, including us, within that group that are not seen within the radially or absent, where symmetry is radial or absent, like the ctenophores, the sponges, the placozoans, and the cnidarians. Okay? These terms, we're going to unpack them as I said today, and you're going to keep bumping into them. So this is important because these are some of the modern tools of a biologist that give you a lot of power that biologists of past generations would have loved to have had access to. Now, some of those tools include this phylogenetic thinking. Some of them include things literally like the microscope that you possess on your phone right now. And when I'm thinking about earlier biologists, I'm going to make a specific reference here to a famous one. And this is a picture of the microscopes at the stage when Charles Darwin was about to set out on the Beagle. And this is a portrait that his family had painted of him before he left. And at the time, as not the eldest son, there was some thought that, confusion within the family. Well, what's Charles going to do? And so giving him passage or garnering for him passage on this ship was a way for the family to kind of help Charles figure out what he was going to do. This is the Darwin that set off on the Beagle. This is the Darwin that came up with the theory of evolution via natural selection. The Darwin that you're more familiar with, that you'll see in a second, this is the one Darwin then had to fight within himself to reconcile his beliefs with the observations that he made in the wild. But a lot of those observations were made with a tool like this. This was a harder microscope to travel with, a harder microscope to have in your pocket. And so this is kind of one measure of the benefits that you have, the obligations and the opportunities, I suppose, that you have as a biologist of this generation. And if you're interested in more about Darwin as he came back from the Beagle, there's a link here that you can scan and go and watch later on. This is Down House, where he lived the majority of his life and where he did a lot of the work. The room, which was well lit, where he did his barnacles, I believe, is through that window right there. And then the garden where he did a lot of his walking and thinking is behind the painter's perspective here and over to the right. And then the worms were kind of everywhere. The Darwin that you're familiar with, that wrote this book that is on the money in the UK, looks like this. But before we leave Darwin in this particular example, this is not the Darwin that did the work. This is the Darwin that kind of fought with writing the book. The Darwin that did the work was this gentleman who is your age. So no pressure on you, but this is what he accomplished at this age, and these are the thoughts. This is the, I suppose, me by comparison saying to you, expect much of yourself. Go out and do these things. Get on that Beagle. Okay, park the advocation, and we'll talk about evolution by natural selection. This is the review. This is the beginning of some review of these terms with a specific invertebrate example so that we all start out on the same page. So VISTA is the acronym for variation, inheritance, selection, time, and adaptation. That's evolution by a natural selection. So unpacking that there are populations within which there is variation, morphological phenotypic variation. That variation can be inherited. That there is what has been called in a very Malthusian kind of way, the overproduction or a lot of offspring. And that through time, there is differential survival and reproduction of those offspring that is determined by the environment. And then finally, there's a result in a change in gene frequencies that correlates with the change in morphology, behavior, physiology, whatever, that is adaptive in the set of environmental conditions, and that is inherited. Right? Do we agree? Now, the example that you've probably been worked or exposed to in the past is with the pepper moth. And this is, I'm claiming it here for the invertebrate world because this is an invertebrate example. These are lepidopterans, they are arthropods. And so the pepper moth, which comes in two forms. There's a dark form and a lighter colored form that if you zoom in and kind of lean in closely, really looks like the edge of or the bark on a birch tree. Now, context, setting the stage. We're thinking about the United Kingdom, and we're thinking about it here on the x-axis through time, from the 1700s to just 2019. And what the y-axis is the production of coal, so millions of tons of coal produced. The first thing that I'm going to ask you to do here is to predict or plot, pause me in a second. I'll give you a timer, you can see where to do it. And show me what you think the production of coal looked like in the UK between 1700 and 2019. This is me pausing. Now you can unpause me, and I'll show you. It basically looks like this. At the 1700, it was not so much, like tens of millions of tons of coal. But then at the start of the Industrial Revolution, this is of course the fuel for the industrialization of the English countryside, of the world at that point. And coal, the production of coal, dramatically increased until it peaks right about there at the 1900s. Now, so many consequences of coal being used to produce steam, being used to drive engines and machines, and one of them is, of course, filthiness. There's clearly a climate, but one of that we're talking about today is just the absolute darkening of the landscape because of the dust. So you're looking at some pictures here from Manchester. Manchester in northern, in the north of the UK, or in the north of England anyhow, grew 10 times. The number of people were just harvested out of the countryside, and it grew in that 100 years about 10, it increased in size by about 10 times. And the skies were dark with the production of these mills. Now, this is the era when William Blake, if you know the poem Jerusalem, when he talked about the dark satanic mills, this is the dark that it's referring to, is the literal like covering of the landscape in this soot. If you're a fan of Tolkien and the Lord of the Rings, at the end, the raising of the Shire, when the orcs and Saruman and company kind of take over the Shire and begin industrializing essentially this bucolic industrial English countryside, that's Tolkien talking about his memories of these industrial furnaces. So covering the landscape with a sheen of a dust of coal, of darkening of coal, changed the frequency of these two forms. With a population within which there is variation. So two forms. The typica, so the scientific name for the pepper moth, Biston betularia. Betularia, if you know about birch at all, this is essentially saying this is the biston that's on the birch. And so typica, this form, looks like birch, the trunk or the bark out of a birch tree. And so this insect could have hidden quite cryptically from its predating bird species. And the carbonara is a darker that would be more easy to spot for a potential predator. Now, I'm going to come up with another timestamp again here, because what I'm going to ask you to do, based on your experience and your familiarity with this example of evolution via natural selection in the invertebrate world, is to take a look at this figure. So we've zoomed in. We're not from the 1700s, but from the 1850s, so from 1840 to 1950, which encompasses that peak, I'll put a little line there, production of coal around 1900. Plot out for me what you would imagine or what you know to be the frequency. So remember, this is going to add up to 1. The frequency in the population of the Typica form and the Carbonara form. And while you do that, I'll have a drink. So I'm paused. All right, unpause me. There it is. So this is, of course, the classicness of this example is that the Typica form, the birch-looking one, was the majority of the population. This was there wasn't a lot of variation, but this is where it was expressed because it was advantageous to be hiding from your predators. Now, as coal dust blanketed the landscape, the trunks of the trees were literally darkened, and these historically well-hidden, lighter-colored individuals were much more easy to eat. So the frequency of the successfully reproducing individuals went down, while the success and the proportion of the population that was comprised of the darker Carbonaria form went up. Okay? So the classicness of this example, let's take a look at it. So we have variation. Does it meet all these VISTA criteria? There is variation in the population, Typica and Carbonaria. That variation is heritable. We're still unpacking as we learn about the genetics of this species how, but the variant for this color seems to be on the 17th chromosome that's responsible for the moth's melanism. There is certainly much production of offspring. That the estimated number of eggs per female moth can be upwards of 350. And then the graph that we unpacked, or the condition that we unpacked most specifically with that graph, was differential survival and reproduction determined by the environment. So increasing dust, increasing frequency of Carbonaria, decreasing frequency of the Typica, the birch-looking one. And so that resulted in the change of gene frequency and yada, yada, yada. So a rush through a beautiful example for which take a look at if you're interested. There is literature. Some of this, there's still two things that are happening. One of them is we're reevaluating some of the early work about the differential bird predation. And we're still figuring out the genetics of melanism. So this is a classic example that you've been introduced to for many, many years that encompasses many of the conditions for our understanding of evolution via natural selection. But there are still things we're learning about it. And then the last aside, as Caramel kind of advises me to change gears here, is to remember, of course, that evolution, as you have learned in other courses and in other exposures, natural selection is only one method by which evolution can take place, but it's a classic one, and so we unpack it here just to share some of these terms and examples together. Now, when we talk about phylogenetic systematics, when we talk about species, remember I said Bistambecularia, we are inevitably bumping into this gentleman, Carl von Linné, a Swedish botanist and zoologist who laid the foundation for modern naming in 260 odd years ago in Sweden. So this is Linnaeus. Kingdom, phylum, class, order, family, genus, species, right? Yeah, right? These are the Linnaean categories. So Bistambecularia is the same as this, the pepper moth that we just went through is an animal, an arthropod, a hexapod. It's a lepidopteran. It's a geometrid, if I remember correctly, and then Bistambecularia. This is the nested hierarchy of another arthropod, in this case, a gorgeous species of yellow-haired ant that is a big carpenter ant in Costa Rica that if you come to Costa Rica, you will see and you will be able to identify as a species. It's kind of a pleasure that way. So Linnaean categories are not the same as phylogenetic systematics or evolutionary thinking. In fact, these categories predate evolutionary thinking. So they're categories that are groupings that are essentially based on observable similarities and differences, and they make up that nested hierarchy: kingdom, phylum, class, order, family, genus, species. And they're typically morphology based, and at least in the 20th century, when old people like me took this class, they were really hammered home. This is, you have to memorize the phylum, you have to memorize the class, you have to memorize the order and all the examples, and then blah, blah, blah. Phylogenetic systematics gives us some more power for navigating the natural world because it's inherently hypothesis-based. And it's tree-based in the way that we're going to use it in this class. So we're talking about groupings based on dichotomous branching, the phylogeny. We're going to emphasize the importance of monophyletic groups. It often uses molecular as well as observable morphological data. And we de-emphasize the Linnaean categories because those are not natural. They're kind of our placing on the world of these rather unnatural things. Now, there was a whole conflict in biology in the last century, 1970s and 80s, when two Canadian biologist brothers, David and Wayne Madison, wrote about this, the cladistic wars, where one camp was the phonetics camp, shown here with almost the Pepsi logo, where biologists believed that just getting more evidence, didn't matter what kind of evidence, more evidence was the better way to understand who was related to whom. Whereas cladistics emphasized that what kind of traits you were looking at really determined how useful and what inference you could make from your phylogeny. And so they caused a little kerfuffle at this Ann Arbor meeting in Michigan in 1981 by wearing these Coke and Pepsi cladistics phonetics t-shirts. You can go read the blog if you're interested in that historical document. And then further, if you're interested in more of a review of tree thinking and its importance in biology, you can take a look at this document here, which is open access, and so just scanning the QR code will take you to that paper. We're going to jump into some of the terms in the remaining time together. Clados, when we talk about phonetics or cladistics is derived from the Greek for branch or clade. And it is what Jan Pechnik has described in one of the textbooks that is on reserve for this course, a terminologically well-endowed field. So there's a lot of terms, and I'm going to talk about them, and then we're going to use some pictures, and you can pause me to kind of share how we identify those kinds of things. So an apomorphy is a derived character or trait of a particular group. A synapomorphy is a shared derived character of two or more taxa. Excuse me. A simplesiomorphy, longest word, just means it's an ancestral trait. And so for a cladist, this is the one where a cladist would say traits that are a shared ancestral trait don't help us differentiate groups, taxa, from each other. They just say that they were related at one point, and that isn't as helpful. So differentiating traits, characters, and character states from simplesiomorphies and synapomorphies is a really important process for a cladist. Now, those are traits, the first three describing characters. Let's focus more so, and we will in this course, on these terms which have to do with looking at those characters on a tree. So a monophyletic taxon, that's one that includes all the descendants of a single immediate ancestor. Paraphyletic is one that includes some, but not all. Polyphyletic, all of the descendants of more than one ancestor. And so we're going to work through some examples of the three of these and then talk about, I'll advocate why monophyly is a useful thing. Differentiating between the three, kind of esoteric, but really being able to identify monophyly or not, that's the power to you as a phylogenetically, systematically informed biology. Because it helps you differentiate these final two categories, which is similarity that is because of shared genetic inheritance, which is a homology or a homologous trait, versus homoplasy, when that's similarity that's essentially described or winnowed away by natural selection because of a strict environmental condition, so convergent evolution. All right, so we have a couple of different trees and some groupings. In gray, you can see, so the taxon W, taxon X, taxon Y, taxon Z. What I would like you to do pause me in a second and show me the taxon that's monophyletic here. Unpause me. All right. You got it. I'm sure you got it. It's W. So that's monophyletic because it includes an ancestor and all of the descendants. So species B, C, and D. The next one, show me, and you're going to pause me again for a second, which taxon here is paraphyletic. So paraphyly, there's one example here with taxon X. Paraphyly is something that includes the ancestor, so like A, but only some of the descendants. So everything E, G, I, H, but it excludes B, C, and D. Taxon Z is also paraphyletic because it excludes M and P. So paraphyly, common ancestor, but not all of the descendant lineages. And then finally, this other category of non-monophyletic grouping, show me which taxon is polyphyletic. Got it? All right. So, and that's, of course, the remainder, that's Y. So M and P. So that's polyphyletic because it is taxa that are not derived from an immediate common ancestor. So N and K. So then why is monophyly useful? Why is Smith advocating for monophyly as a principal mechanism for the strength of phylogenetic systematics to accelerate our understanding of the invertebrate world, which is the animal world at large? And that's because essentially it has to do with missing information. So missing information, however you analyze, whatever you're analyzing, creates problems, right? Whether it's genealogical or evolutionary, all of these things. A paraphyletic group, one that excludes things, is missing information. And so your understanding of that group is plagued by this missing information. So in evolutionary terms, this monophyletic group, this is a real biological unit. So it's the product of descent with modification. So an ancestor and all of the descendants. And as such, you can use it to study the evolutionary processes that produce them or make predictions because one of the themes already, we're 2 lectures in and Smith is talking about themes, is that there are many more things to discover than we have already discovered. This is the unknown unknowns, the Jon Snow. We are the Jon Snows of the animal biological world, that we're trying to understand all these species that we have not named yet. And so as species are discovered, their placement, their phylogenetic placement, is essentially us making a hypothesis about membership, shared membership in a monophyletic group. And if we do that well, using these properly kind of measured traits, that lets us predict things about that we might not yet know, we might not yet have a chance to have studied for these novel taxa that we would then place within a monophyletic group. Paraphyletic, polyphyletic groups, these are essentially, so you could think of them as being the source of human error. Excuse me. Incomplete information, flawed information, poor description of characters, or a poor understanding of how those characters are actually used by the organism in question. And I'll give you I think it's a really cool example in a second about gelatinous organisms. So using a para or a polyphyletic group to study evolutionary processes directs us along misleading or confusing pathways. And this is because it essentially is misplacing. One of the errors could be, well, if there's a trait that we ascribe to shared or common inheritance that has actually come about because of a strict environmental condition that has led to the similarity that we're identifying via convergent evolution, that we're missing the opportunity to understand how the environment is affecting the evolutionary process of that organism, or the rebound, how we might learn from that trait to modify our own behavior or technology and move forward in an energetically efficient way. So, some more pausing here. I'm going to ask, so this is the, I've been rotating. This is the done. We've returned to the done et al. Phylogeny, and instead of being vertical, it's now dripping down. It's hanging like icicles, okay? So the Spiralia that I talked about earlier that contains mollusks, etc., that's here. So that's that clade. So what I want you to do now on your own, pause me for a second, because we're going to talk about, I'm going to ask you to look for 3 different things. I'm also going to pause myself to say I've used this word already in Monday's lecture and today, a taxon or a taxa. So this is, I can talk about any part of a Linnaean hierarchy, like so a phylum, the ctenophores, that's a taxon. Ctenophores and periferans, those are taxa, 2 different taxa. But we can talk about them, species are taxa, phyla are taxa, but generally taxa should be used, when used usefully, they're referring to monophyletic groups. Okay? Just some shared agreement about terms as we go on. So what I want you to do, when you pause me in a second, is show me the root of the phylogeny, show me there's a polytomy, where it's an unresolved, and show me the tips. All right, there's 10 seconds. So I'm unpaused, and you probably found the root, right? So the root is the base of the tree. The root is a portion that generally is described by your in-group and your out-group. We'll talk about the out-group in a second. The out-group here, since these are all the animals, would be the choanoflagellates. That's a bit of a, we're jumping ahead, but so you've seen it already in Monday's lecture, and we'll talk about it in Friday's lecture. So some other, so the tips of the tree, so like the urochordates or the craniates, these are tips. This is our clade here. But these are other things, other measures of a tree. So there's a monophyletic group there with the panarthropods, with tardigrades, onychophorans, and arthropods. A polytomy, so an unresolved and non-dichotomous branching pattern where the relationships between annelids, mollusks, nemerteans, brachiopods, and pharonids is unresolved. The spiralia itself is another one. The length of these branches, of course, is shown here. And then we can plot onto this 2 different, we can plot on names, so like this U shape or upside down U that I'll talk about in a second, or we can plot on audapomorphies or the traits. And so comb rows is a trait that is specific to the ctenophore. So we could put a ctenophore trait right there. Now this grouping, this U-shaped grouping, would show you a non-monophyletic group that we actually, that I was taught about, called the Coelenterata. Now the Coelenterata is essentially gelatinous organisms in the ocean. And it was a grouping that historically, that I was taught about between the cnidarians and the ctenophores. And cnidarians are radially symmetrical, ctenophores aren't, but they're both gelatinous and they're both hard to observe. And cnidarians tend to have stinging cells, and ctenophores have coloblasts or sticky cells, but we kind of put them together until this paper that came out of the University of Victoria and included a graduate student. So Mills, the lead author, was a graduate student at the time, who talked about the fact that the majority of times that we thought there were stinging cells in the ctenophores, so the characteristic, the synapomorphy of the cnidarians, but occurring in the ctenophores, it was always because ctenophores had actually eaten cnidarians. So they didn't have this trait. It wasn't an infrequent trait that some groups had lost, because evolution is not just the accumulation of things, it's often the loss of these traits. We'll talk about lots and lots of examples of this. So this was this graduate student who said to the world, this is kind of a big deal, right? This is a giant group and describes most of, if you were an alien crash landing onto the Earth, you'd wonder why we called it the Earth and not the water. because the majority of where that ship would crash is into the water, and probably the first life that they would bump into is gelatinous. That's what fills up our ocean. And so this is a graduate student who did a major reshuffling about how we understood. So the Coelenterata, this artificial, non-monophyletic group between these distantly related but gelatinous, superficially similar looking things, is not a monophyletic group. So I put it in the quotes. And what you'll hear me talk about it in the course is as a Tribbiani group. So we used to think this, but now it's no longer useful. So the outgroup in that phylogeny would have been the choanoflagellates. The outgroup is a close relative of the members to your in-group, so it's described relationally, and that provides the basis for the comparison. Now, generally, of course, your outgroup is not ancestral. It is as old a lineage as whatever is in your in-group, but the characteristics that it has are assumed to represent the ancestral condition. But it's an important thing to recognize that those tips, if you're plotting a tree that has extant tips, still living tips, they're the same age. Okay, so this cladistic analysis, this kind of phylogenetic systematics, is about, and this is a small line about these derived characters, that we want them to be not ancestral. And when you're building a tree, and there's all sorts of statistical ways to build a tree, cladistics is just one of a whole quiver of tools that phylogenetic systematists have to do with it. There are multiple trees that are possible. So all these trees are essentially hypotheses that you support or don't support with new information that you collect. And we tend to use this phrase, Occam's razor, where the simplest explanation is the preferable one, to help us choose between trees. I'll give you a small bit of math here, or an example where if we have three different kind of invertebrate little weirdo organisms here, imaginary organisms, who evolved first? How many answers are there? Many. This is the math to describe how many possible. So if we increase the size of the tree, and you can see there are a lot of potential answers. And so choosing between, we have this rule of thumb that the simplest one, we use Occam's razor, keep calm and use Occam's razor, that the simplest explanation is the preferred one. But of course, remember that itself is an assumption, that it often leads us in the right direction, but there's no evolutionary rule or physical rule of the world to describe that this is always how evolution would take place. It's often how evolution takes place. So this is a quick run through shared terms and a common example that sets the stage for us to phylogenetically consider all of these taxa within the protozoan and invertebrate world. The eukaryotes. So we're going to have a quick shared history here with eukaryotes. So that phrase eukaryotes, is the presence of a nucleus, so the true, and the karyo refers to a kernel or seed, which is a description of the nucleus. And we differentiate it from the typical prokaryotic cell, which is a fairly simple bacterial thing here, no true nucleus, versus this classic thing that if you, grew up in a small rural area, maybe you made one of these out of Jell-O at a certain point in your training, but it's membrane-bound organelles, right? You've got the mitochondria. Does everybody hear it? Powerhouse of the cell. Now, how do we, from an evolutionary perspective, how did animal diversity proceed from vast, successful, prokaryotic world, where still the majority of species are likely prokaryotic species, but where did the animals come from when this was the world for many, many millions of years? And one of the theories about how that proceeded was called the serial endosymbiotic theory. So we have this number one here on the left-hand side of the screen, this anaerobic heterotrophic prokaryote. So it's kind of moving around, bumping into things and eating things. And then we see this consuming but not digestion of this aerobic heterotroph, this other kind of prokaryote that becomes the proto-mitochondria. And then if you are a plant or something that's photosynthesizing, there's a secondary event where there's even a third, pardon me, a consumption without digestion. And then through time, this phytoflagellate, so there's a nucleus and the organelle structure, so a plastid and a mitochondria, caused by this chimera, caused by this serial, so subsequent, one then the other, endosymbiosis, so consumption of smaller things. Now, as a Guelph student, you're familiar, of course, with chimeras because your logo, you are griffins. A griffin is a chimera. What is the kind of evidence that we have for the serial endosymbiotic theory? A lot of it has to do with this. You're going to see multiple examples here, but one of the important ones is that mitochondria and chloroplasts have their own DNA and replicate themselves, and they are dividing and replicating at a different rate and very rarely interchanging directly with the nucleus. Strongly suggests a different evolutionary history. This is added to by the fact that the mitochondria has a different genetic code. There are double membranes surrounding these. There's also RNA similarities between extant bacteria and mitochondrial DNA and chloroplasts of extant eukaryotes. And that we've seen recent endosymbiotic events in protists. So we can model some just so stories, these partial digestions and maintenance, kind of interruptions of digestion and then and maintenance. So facultative endosymbionts. So I'll give you an example here. So this is some recent evidence for the serial endosymbiotic theory. And this was, so we've got a bacteria, or this Mesodynium chameleon, this food vacuole, so the creation of a food vacuole, so endosymbiosis, it reaches out and eats something, and then it acts as there's a benefit towards the consumer because that which it has consumed, the plastid containing organism, the algal cell, continues to act as a photosynthetic unit. And so the recently digested algal cell adds to the consumer. And so there is some older, so here's a 1 and 2 and 3, so the algae is eventually digested. But in seeing these contemporary examples, this adds weight to the principal forms of support for the serial endosymbiotic theory, which come from the independence of the DNA structurally, and then the independence translationally, and then these modern examples. And so this area, like, what if we could review and renew the tape of life? This is a continuing area of active inquiry, of scientific inquiry. There's a philosophical transitions as a example that there's a whole special unit that's devoted to the origin of eukaryotes, and that it is a fundamental, forbidding evolutionary puzzle. All right, so we've been through a bunch together already. This is Wednesday. Tranquilo. Friday, we're going to rejoin. We're going to begin to talk about some of the protozoan lineages that will underpin or kind of lead us into thinking about the metazoan eukaryotes. So today we talked about evolution by natural selection. We reclaimed this invertebrate example. We shared our understanding of useful terms of phylogenetic systematics and Linnaean categories and monophyly, the utility of monophyly, the utility of tree-based thinking. And we talked a little bit about serial endosymbiotic theory and the origin of eukaryotes. So I'm going to stop recording and wish you the best of the middle of this week. Take care of yourself, take care of those around you, and we'll see you on Friday morning.